Hi viewers, this is J. Swamy, Assistant Professor of Zoology. Today we are going to discuss absorption and assimilation of digested food. Under this topic we will discuss introduction to the absorption and assimilation, anatomy of villi and microvilli of small intestine, methods of absorption, absorption of carbohydrates, absorption of proteins, absorption of lipids, absorption of other substances like the water and vitamins, and finally the assimilation. See the introductory part. The main aim of the digestion is the conversion of complex substances into the simple substances which are assimilable or absorbable by the cells. That is, each and every cell of our body should be supplied with digested food. This is very essential. That is, contribution of digested food all the cells is very essential for the growth, energy production and the metabolical activities. This is only possible when the absorption and the assimilation of the food material has been taken place. In the process of absorption, the digested food that should reach the small intestine, and that is the epithelial cells of the small intestine, and thereby to the blood vessels or the lacteals, and all the blood vessels which are present over the small intestine, they fuse to form hepatic portal vein that reaches the liver and finally mixed up with the main circulatory system, and through the circulatory system all the food material supplied to all the kinds of cells and tissues. Now in the assimilation process, digested food that reaches the cells and tissues and that will be utilized by the cells, carbohydrates for the energy production and amino acids for the construction of the lipids, that is construction of the proteins, that is the protein synthesis, and so on. Now in order to reach the circulation, the food material, digested food material has to cross two barriers. One is the, let us see the structure of the villi of the small intestine. It has to cross, for example, the food material is here. This is the lumen of the small intestine. Now, in order to reach the circulation, it has to cross the epithelial membrane of the small intestine and it has to cross the endothelial membrane of the blood vessels. Endothelial membrane of the blood vessel. Then only it has to reach the circulatory system. That is, the food material, in order to reach the circulatory system, circulation, it has to cross two barriers. One of that is epithelial cells of the intestine, small intestine, and second one is the endothelial cells of the blood capillaries. Of about 90% of the nutrients get absorbed by the small intestine, whereas only 10% like some of the drugs and alcohol, they may be absorbed in various parts of the alimentary canal, but 90% of the food material get absorbed by the small intestine only. And here these are the villi of the intestine. All these are, so in the section they will look like this, and at the end of the at the tip of the villi we will see a brush border. Here it is clearly visible. These are the microvilli of the intestine villi. Villi, it is a finger-like projection and it again subdivided into microvilli. Okay, they will form a brush border. And due to the presence of the microvilli and villi in a specific arrangement, the inner side small intestine surface area may be enhanced, otherwise may be lengthens of about 300 square meters. Due to the specific arrangement of the villi and microvilli, the inner side volume of the small intestine, it is of about 300 square meters. It is almost all equal to a tennis court. And this kind of arrangement and as well as the microvilli, they enhances the area of absorption. They enhances the absorption area within the small intestine, that we should remember. And if you see furthermore, each and every villi, we will see blood vessels. This red one is the artery and blue one is the vein. And here it is a green color thing, this is the lacteal. So lymph vessel, lacteal means lymph vessel. Okay, each and every villi consisting of blood vessels and the lacteal vessel, otherwise lymph vessel. Why there is a deviation? Here it is, out of all the digested food, monosaccharides and amino acids, they reach blood circulation. Amino acids and the monosaccharides, they enter into blood circulation, whereas fatty acids and glycerol, they enter into lacteal vessel. They enter into lacteal vessel. Each and every microvilli consist of blood vessels and the lacteal vessels. Monosaccharides and amino acids absorbed into the blood vessels, whereas the fatty acids and glycerol absorbed into the lacteals. This is the fundamental rule, right? And you see the routes of transportation of the food material. There are basically two routes. One is the hepatic portal system, another one is the lymphatic system. Hepatic portal system and the lymphatic system. What is the hepatic? What is the portal system? Actually, the small capillaries which are present in a particular organ, they fuse it to form, for example, like this. So these are the small capillaries, they fuse it to form a large vessel. They reach another organ and again subdivide into small capillaries. The capillaries which are started from a organ, that will fuse it to form a large vessel, and that will subdivided into capillaries again in another organ, that is the hepatic portal system. If that starts in the alimentary canal, ends in the liver, that is the hepatic portal system. If that is starts at the hind limbs and tail, that will ends with the kidneys, that is the renal portal system. So here hepatic portal system is present. In the hepatic portal system, that starts with the small intestine and ends with the liver, that carries the absorbed food material into the liver. In the lymphatic system, there are two types of fluid connective tissues in our body. One is the blood, another one is the lymph, right? The lymphatic system also, it collects the fatty acids and the glycerol, and that reaches to the thoracic region and finally end up with the main circulatory system. So these are the two routes of transportation of the digested food. And see the methods of digestion. Here basically four kinds of absorption methods are there, namely diffusion, osmosis, active transport and the facilitated diffusion. Diffusion is a process where there is a movement of the substances or molecules from higher concentration level to the lower concentration level according to the concentration gradient is the diffusion. Osmosis is a process where there is a movement of molecules from lower concentration to the higher concentration against to the concentration gradient through the semi-permeable membranes. And coming to the third one, active transport. Active transport is the transport where for the transportation it utilizes energy. If the active transport utilizes the energy from the source of ATP, this is known as the primary active transport. Primary active transport. Whereas if they use the electrochemical gradient, then that is known as the secondary active transport. Active transport is a transport with the utilization of the energy. If they use energy from the source of ATP, primary active transport. And if they utilize the electrochemical gradient energy, then that is secondary active transport. The best example for the secondary active transport is the absorption of the glucose. Then coming to the fourth one, facilitated diffusion. So here the movement of the molecules from higher concentration level to lower concentration level takes place, but with the help of some carrier molecules. That is known as facilitated diffusion. Then come to the how the absorption of carbohydrates takes place in the small intestine. As we know, due to as a result of the carbohydrate digestion that gives rise to the formation of monosaccharides like glucose, fructose and galactose. And the sugars, they will be absorbed in two methods. The monosaccharides like glucose and galactose, they transport or absorbed through the secondary active transport method. Just now we discussed. Secondary active transport method is a method where there is a active transport by utilization of the electrochemical gradient of the cell membrane, right? And the fructose exhibits fructose absorbed by the facilitated diffusion, glucose, galactose by the secondary active transport, and the fructose by the facilitated diffusion. Glucose along with the sodium ions, they may be co-transported through the epithelial membrane of the intestine. They may also use the carrier proteins. In the process of co-transportation, right? And there is a sodium potassium ATPase pump which always allows the 3 sodium ions inside to the circulation and 2 potassium ions that will sent out from the circulation to the cells. Sodium potassium ATPase pump, right? And then continuously there will be a co-transportation of glucose and sodium. Glucose transportation may be inhibited by various drugs like ovabane and cardiac glycoside and fluorigine. Ovabane and cardiac glycoside and the fluorigine, right? Here we will see glucose and galactose, they are transported through secondary active transport mechanism, and whereas the fructose, that is with the facilitated diffusion, and glucose and sodium, they may be transported through the mode of co-transport, and there is a sodium potassium ATPase pump. 3 sodium molecules get inside and 2 potassium molecules exit outside, and glucose enter into the blood through the facilitated diffusion. So these are the methods of the absorption of the carbohydrates. The glucose transport may be inhibited by the drugs like ovabane, cardiac glycoside and fluorigine. Then if you observe the image, here there is a glucose and sodium, they entered into the cell. Enterocyte is the cell of small intestine, and again that is every digested food like the glucose, it has to cross the epithelial membrane of the small intestine and the endothelial walls of the blood vessel. Okay. Now in the first step, what happens? The glucose enters into the cell and again enters into the circulation. Galactose, same, and the fructose. So the methods are different, right? And here it is a sodium potassium ATPase pump. 3 sodium molecules enters into the circulation and 2 potassium molecules exit from the circulation, right? Sodium potassium ATPase is the enzyme. Sodium potassium ATPase is the enzyme. Okay, there is a special channel through which sodium and potassium ions get enter and exit from the circulation. This is about the absorption of the carbohydrates. And coming to the absorption of the amino acids, proteins. How the proteins get absorbed, right? Basically, the digestion of proteins results in the formation of amino acids, which are the building blocks of the proteins, and these amino acids, dipeptides and tripeptides, they enter into the small intestine. Basically, levorotatory amino acids, L amino acids, they absorbed through the secondary active transport, that is, they utilize electrochemical gradient of the cell membrane for their transport. Then dipeptides and tripeptides, they also follow the secondary active transport, but they are always mixed up with the protons, H plus ions. Along with the H plus, they are absorbed into the small intestine. Here, these are the small peptides along with the hydrogen, they enter inside. Amino acids, some of them along with the sodium, and here some large peptides also enters into the. So these all are the cells of small intestine, and this is the circulation. So they have to cross, amino acids also cross epithelial cells of the small intestine and endothelial cells of the blood vessels. Then see the absorption of the lipids. It is somewhat critical. As a result of the digestion of lipids, that leads to the formation of fatty acids, glycerol and monoglycerides, diglycerides, monoglycerides and the diglycerides. All of them have to be absorbed into the small intestine, right? Now, there are certain conditions which are the lipids which we have ingested into our body, they should not be absorbed, they should not be transported in their native form. This is the first and fundamental thing. Lipids in their native form neither absorbed nor transported, so that they have to be modified into two forms, namely either into the micelles or into the chilomicrons. These two things to be absorbed by the cells, right? It has to be taken place in two phases. In the first phase, what happens? Monoglycerides, fatty acids, they mixed up with the bile salts, phospholipids, cholesterol and the fat soluble vitamins. All of them they fuse to form the micelle, small droplet like structures. They will be released into or absorbed into the enterocytes, that is the epithelial cells of the small intestine, through diffusion or through the help of the carrier proteins. This is the first phase, and they enter into the endoplasmic reticulum of the enterocytes, and again they will be resynthesized into the triacyl glycerol, again into the fat. Understood? In the first phase, what happens? All the monoglycerides, fatty acids, that is they mix up with the bile salts, phospholipids, cholesterol and fat soluble vitamins, and finally they form into small oil droplet like structures. They will reach the small intestine, otherwise enterocytes of the small intestine, through the diffusion or through the help of the carrier proteins. And after reaching into the enterocytes, they enter into the endoplasmic reticulum and again resynthesize into triacylglycerol. This is the first phase. See, lipids do not absorb in their native form. Lipids modified to micelles and chilomicrons. So this happens in two phases. In the first phase, the formation of micelle takes place with the help of the bile salts, phospholipids, cholesterol and the fat soluble vitamins. In the second phase, what happens? Triglycerides, they mix up with the cholesterol and fat soluble vitamins, and they are surrounded by lipoprotein layer or protein layer, and they become small granular forms. In the second phase, triglycerides mixed up with the cholesterol and the fat soluble vitamins, surrounded by a protein membrane and converted into small granular forms. These small granular forms are nothing but the chilomicrons. These are surrounded by lipoprotein layer and easily transported through the circulation. These chilomicrons, they come out from the enterocytes through the process of exocytosis. And they reach into lacteals. So while discussing this topic, in the initially we have came to know that is monosaccharides and the amino acids, they transported through the circulation. They enter into the blood vessels in the small intestine, whereas the fatty acids and glycerol, they enter into the lacteals. So after the formation of the chilomicrons, they enter into the lacteals through the process of exocytosis. Now, thereby they will reach the lymph vessel and finally mix up with the blood at the neck region, and during the transportation through the blood vessel, the endothelial cells of the blood vessel, they release a certain enzyme which is known as the lipoprotein lipase. Right? The absorbed chilomicrons, as and when they reach the circulation from the lacteals, during the transportation within the circulatory system within the blood vessel, the endothelial cells of the blood vessel, they release a particular enzyme which is known as the lipoprotein lipase. What is the function of the lipoprotein lipase is it can cleave or digest the lipoprotein layer of the chilomicrons, and thereby the monoglycerides, fatty acids, they will be released freely into the cells. Simply, what happens in the absorption of the lipids? In their native form, they may not absorb. They have to modify into either the micelle or the chilomicrons. First phase is the formation of micelle, right? And the second phase is the formation of chilomicrons. And finally, these chilomicrons, they enter into the circulation through the exocytosis, and thereby the lipoprotein layer which is surrounded the chilomicrons, that is get digested by the lipoprotein lipase enzyme which is released by the endothelial cells of the blood vessel, and thereby the fatty acids and the monoglycerides, all of them, they are released freely into the cells and they reach host cells. Right? This is about the absorption of the fatty acids. Right? Here what happens, this is the large lipid molecule, it undergoes emulsification and formed into the micelles. Okay, it enters into the enterocytes and the endomembrane system that forms the chilomicrons. And these chilomicrons, they enter into the lacteals and finally lacteals to the circulation. Right? Then absorption of the other substances like the water and vitamins. The water absorbed in all the regions of the alimentary canal like the mouth, stomach, small intestine and the large intestine. But most of the water get absorbed by the large intestine through the process of osmosis. And the water soluble vitamins like the B and C vitamins, they also absorbed along with the water. Whereas the fat soluble vitamins, they will absorbed and transported along with the glycerol and the fatty acids. Glycerol and the fatty acids. Right? And coming to the overview, here we will see the pink color ones, amino acids, carbohydrates, long chain fatty acids, short chain fatty acids, right? All of them, they reach the villi. In the villi, amino acids and carbohydrates enters into the blood vessel, whereas the fatty acids, glycerol enter into the lacteals. So these blood vessels, they form the hepatic portal vein and reaches the liver, right? And if at all it is necessary, they mix up with the blood circulation. The lacteals which absorbed the fatty acids and glycerol, they also mix up with the, finally they mix up the lymph vessels, comes in contact with the blood vessels at the neck region, right? And the water as usually that will be absorbed in all portions of the alimentary canal. Now, see the assimilation process. The digested food in the form of monosaccharides, amino acids, fatty acids and the glycerol. What is the assimilation is utilization of the digested food, utilization of the absorbed food. As and when the digested food reach each and every cell, that should be utilized by the cell for its metabolical activities, for the production of energy, for the generation of energy or for the growth of the individual. See, the amino acids, they are meant for the, they utilized for the protein synthesis. Already we discussed during the protein digestion, each and every organism require its own proteins. It synthesizes its own proteins. These proteins are not at all useful for the other organisms. So that they should be get divided into amino acids and they form their own proteins based upon their needs and necessities and requirements. So that the amino acids which are entered into the cell, they go for the protein synthesis. And monosaccharides meant for the generation of ATP through the glycolysis, Krebs cycle and electron transport system. And the extra sugars, they will be converted into glycogen and stored in the liver and as well as the muscles through the process of glucogenesis. Fatty acids and the glycerol, they again formed into fats and they stored in the adipose tissue which is located near the subcutaneous membrane, subcutaneous region of the skin. So that is various ingredients of the digested food like the monosaccharides, amino acids and the fatty acids and glycerol, they will be highly utilized for the needs and necessities of the organism's body, for the growth, generation of energy and for running of metabolical activities and the generation of energy and so on. So this is all about the assimilation of the food material. Thank you one and all.